So we know a lot about the details, and so I want at the level of single cell, but I want to go now to the network, and a particular network I just mentioned, the cortical column. So this column that looks like, in this case, a barrel, this column is very specific and anatomically well defined in the case of the mouse whisker system. We call it the barrel cortex. So in the case of the barrel cortex, we know that in the cortex, in particular somatosensory cortex of the mouse, there are these barrels. So this is the surface of the cortex. This is the depth, 2 millimeter deep cortex. In this region, there are these columns that each represent information, primarily information flowing from a particular whisker. So this whisker, the sensing of this whisker, the activity, the information coming from this whisker may flow and eventually goes into a particular column, a group of 10,000, 20,000 cells. We may look at it like this. So this is the whisker system, one whisker, another whisker of the mouse. Okay? And each whisker has a name because it is repetitive. So this is one whisker, another whisker on the surface of the face here. And you can see that there is a very clear mapping between the location of a particular whisker and a particular barrel, a particular column in the cortex of the mouse. So this whisker is represented here. This other whisker is represented here, column by column, nearby anatomically. So here, in the case of the whisker system of the barrel cortex of the mouse, the notion of column is very, very clear. Many people today are working on trying to understand the columnar organization or the columnar representation of whisker information onto the cortex. It's a very nice system because of the clear mapping between a whisker and a particular activity in a column. Of course, eventually the columns interact with each other, so this activity spreads by primarily this particular whisker. When you move it, when you just touch something here, it would ignite primarily a particular column, a particular barrel. And people like Bert Sakmann, Nobel laureate, and many others are working on this, again trying to take a column, a particular column from the whisker system, and try to characterize the cell types. So you see, for example, we just discussed layer 5. These are the big, beautiful cortical pyramidal cell in the barrel cortex. You see this big dendritic tree. You see spiny stellate cells and others in layer 4 and so on. So you can really look at the different cell types at the different layers that I mentioned before. Layer 1, layer 2, 3, layer and so on. So also in the barrel cortex, as in other cortices, typically you have 6 layers. Typically you have all these different cell types with different combinations of cell types. And in the case of Bert Sakmann in his lab, in his group, they're also working on trying to characterize the input coming from the thalamus. So here is an axon, reconstructed axon, coming from the thalamus into the cortex. You can see a very dense termination in layer 4. So this is layer 4. You see big axonal ramification in this region, so dense in this region, but also makes contact other regions. So this input coming from the thalamus is talking to all layers, including layer 2, but not layer 1. So this is an anatomical characterization of the input from the thalamus to this cortex. So this is a column. So when I'm saying a column, in the case of the whisker system, of the barrel cortex of the mouse, the column is anatomically very clear. And also functionally, at least in the very beginning, because you know that early times, after you touch the whisker, this particular column will be ignited. We can also do other anatomical things. This is coming from the work of Henry Markram in another somatosensory system of the mouse. in this case of the rat actually, you can now characterize using new techniques all the excitatory cells in green, all the neurons in green now, all the inhibitory neurons in red, so you can start to now, in a systematic way, map different cell types to different regions into a column that is functionally relevant, at least in the case of the mouse, of the somatosensory cortex, because you know that the information is mostly from a particular whisker. What are the other properties of a column in other systems? So I mentioned before that when you move a line, a bar, continuously, when you move a bar, you know that this bar is oriented in this direction and that it moved from here to here. So this is now a visual input. It's not a somatosensory input. And the question is, so what happens in your visual brain, in your V1, in your early visual system, when you see and you recognize a bar moving? We mentioned Hubel and Wiesel work. We mentioned that when you record in a cell in V1 of a cat, this is also true for the monkey and also for the mouse. When you record in this region, you see that in particular orientation, this cell tends to fire spikes. But what happens to the whole network? So this is a beautiful work from Clay Reed's lab in Harvard, whereby he uses this optical imaging, these calcium-sensitive dyes in cat V1, and then you discover something that was known already from Hubel and Wiesel, that in the cat V1, you have a functional column whereby the whole column, the whole cubic 2 millimeter, tend to be sensitive to a particular orientation. So you can see that if you record from many, many cells with this 2-photon imaging, and each color now codes for a direction, so red means going from up downward, and the other color, for example, this orange color means going this direction, and so on. You can see that a column of many, many cells, all of them tend to behave similarly. They have the same orientation tuning. They have the same directional selectivity. So here is a column in terms of function. There are no this clear splitting of columns anatomically in V1 of the cat, but functionally, as Hubel and Wiesel already described, functionally a column can be characterized by the response of its cells to a particular visual input. So you can summarize what we know about the visual input, the visual structure in the V1 of the cat in this case as follows. You see that there are these functional columns. So this is a functional column whereby all the cells in this column like to respond to this orientation. In other column, to this orientation. You can see that this orientation gradually changes between one column to another column. So this is orientation columns. Functionally, the whole column responds. All the cells tend to respond mostly to this orientation. Not all the cells, but many of the cells. Also, you can see that there is another systematic ordering in V1, because you see that a particular column likes to respond to input from left eye, and then the next one into this axis responds to input from the right ear, and so forth. This is called the ice cube model of the visual cortex of the cat. So I want to speak about columns because the column, the cortical column, is our target tissue for the blue brain simulation. Note also, and that's very important, that not only that the column is a functional unit in many ways, also anatomical unit in the case of the barrel cortex of the whisker, but this column is also plastic. As neurons are plastic, synapses are plastic, the column is changing plastically depending on the early experience in life, and maybe later on, of the animal. For example, from set of very beautiful very beautiful experiments, you can show that if the animal, the cat, grows in an environment with the limited angles, visual angles in the world, let's say only 45 degrees, then all the cortical columns will behave, will respond only to this angle. Just because the animal did not experience at a critical period of time the whole range, the whole world of angles, of visual angles. So this column is not a static tissue. It's a dynamical plastic tissue that may change depending on experience. And of course, all these ingredients you want to capture in your model. So let's go now and discuss the Blue Brain project, trying to simulate mathematically in fine details what we know about anatomy and physiology of a cortical column, a generic cortical column. So as I said before, we know a lot about cell types. We know a lot about the firing properties, spiking properties of the cells. We know quite a lot, not completely, about connectivity, anatomy of dendrites, synaptic properties and dynamics, but we don't understand really how these ingredients generate together a dynamical functional response. We need to integrate this data at some level of resolution and try to understand how the global dynamical property of a whole network emerges from the properties of the individuals, the synapses, the spikes, and so on. So let's see what we do.